Alright, ji, assalamu alaikum everybody. How are you all? I hope that you all are doing well. Today we will be looking at AS and A level biology paper 1-2, which is of October-November 2024, and this paper is of 1 hour and 15 minutes. Let's start with question number 1 over here. Question number 1 over here says that a light microscope is used to observe two structures that are 200 nanometers apart on the slide. What is the actual distance between the two structures when the magnification is changed from into 40 to into 400? Now magnification का हमारे पास formula होता है image length divided by the actual length. अब यहाँ पे यू हैव टू अंडरस्टैंड कि मैग्नीफिकेशन हमारे पास क्या करता है, इट इंक्रीजेज और डिक्रीजेज दी माइक्रोग्राफ, राइट. इट इंक्रीजेज और डिक्रीजेज दी स्ट्रक्चर, increases or decreases the size of the structure. Alright, so it will not affect the actual distance. The magnification, whether the magnification is into 40 or into 200, the actual distance will basically remain the same. So the answer over here would be option C, 200 nanometers. Alright, so now let's move on to question number 2 over here. Let's see that what they are asking us in this question. A cell is shown in the micrograph. Alright, so this is a micrograph given to you, and then they are saying that which statement explains how it is possible to identify the type of microscope used to produce the micrograph. Alright, so we can clearly over here see that these structures, let me highlight them. These structures are the rough endoplasmic reticulum. As if you zoom in this and you see it more closely, you guys can see small dots over here which are of the ribosomes, right? So this is basically the rough endoplasmic reticulum over here, which has the ribosomes as well. And we know that the rough endoplasmic reticulum is visible only in the electron micrograph, right? So over here we can say that option D over here is our correct answer, that ribosomes are visible, so an electron microscope was used. Also, in additional knowledge, if we talk about that what is this structure, so basically these structures are the secretory vesicles. Alright, so the answer over here is option D. Let's now move on to question number 3. Question number 3 over here is saying that which statement supports the fact that mature plant cells contain organelles that carry out the same role as lysosomes. Lysosomes are basically the single membrane bound organelles, and they arise from the Golgi apparatus. They arise from the Golgi apparatus, and they contain hydrolytic enzymes which carry out the hydrolysis, and they contain the hydrogen peroxide which is toxic to the cell. Alright, so the answer can be that a range of hydrolytic enzymes can be found within the mature plant vacuole. Yes, so the answer can be option A, because over here we can see that the hydrolytic enzymes are being found in the mature plant vacuole, which is also found in the lysosomes. So hence the answer over here is option A. Alright, so now let's move on to question 4. It over here says that which cell structures may contain cisternae. There is one term which is known as the cisternae, and there is the other term which is known as the crista, या फिर we can say the cristae. Cristae are basically the folds of the inner membrane of the mitochondria, you can say, or the chloroplast, you can say, because both of these structures, be it the mitochondria or be it the chloroplast, they both have the double membrane, right? The outer membrane and the inner membrane. So basically what are the cristae? Cristae are basically the folds of the inner membrane, like these folds. They are basically called the cristae, and they are found in the mitochondria and the chloroplast. And over here in this question they are talking about cisternae. Cisternae are basically found in the endoplasmic reticulum and the Golgi bodies. We can draw the structure of the Golgi body for better understanding. Like these are the Golgi body, and so they are basically the stacks of the curved flattened sacs जिसे हम लोग पाउचेस वगैरह भी बोलते हैं, एंड इट इज़ ऑल्सो नोन एज सिस्टर्नाई. ऑलराइट, so now let's move. So the answer to this question over here is option C, that the cisternae are basically found in the endoplasmic reticulum and the Golgi body, and they are not found in the mitochondria or the chloroplast. Now let's move on to question number 5 over here. Question number 5 over here says that the diagram shows some cell structures of one type of a cell. Alright, so as per the structure given, we can clearly identify that it is a bacterial cell, or we can say that it is a prokaryotic cell. Over here in this question, they are saying that which labeled cell structures are present in a typical eukaryotic cell, which is an animal cell, and a typical bacterial cell. Now, in order to solve this question, we must first identify the structures 1, 2, 3, 4 and 5 given. Structure 1, you guys can see over here, is basically pointing to the cell membrane. It is pointing towards the cell membrane. The structure 2 is basically pointing towards the slimy capsule. The structure 3 is the bacterial DNA, or we can say that it is just a DNA. Structure 4 is the ribosomes, and 70S, specifically 70S ribosomes are found in the bacterial cells, and structure 5 is the cytoplasm, right, in which all the cell organelles are contained. Now they are saying that which structure is common to both the bacterial cell and the eukaryotic cells, right? So let's identify. Cell membrane is a common cell structure. Slimy capsule is not found in the eukaryotic cells. DNA is found in the eukaryotic cells. Ribosomes are found in the eukaryotic cell, and cytoplasm is also found in the eukaryotic cell. So only structure 2 is not found in the eukaryotic cells. So hence the answer over here would be option B, 1, 3, 4 and 5. Option B is our correct answer. I hope this MCQ is clear to all of you. Now let's move on to question number 6 over here. Question number 6 over here is saying that the diagram shows part of a collagen fibril made of the collagen triple helices. The collagen triple helices are linked to each other by one type of the bond. This bond is labeled as X in the diagram. So basically over here, which bond they are talking about is this bond, right? From which the collagen triple helix structure is, two collagen triple helix structures are joined together, right? So they are basically asking that what is the bond X. So सबसे पहले let's identify collagen. Collagen is basically collagen. What is collagen? Collagen is a protein, and it is a, if we get into the classification of the protein, so basically collagen is a fibrous protein, right? Collagen is a fibrous protein, and the structures which provide the stability and cross linkage to the collagen is basically the covalent bond. So hence the answer over here is option A, covalent bonds. If you look, and you can understand this by looking at the structure of the collagen and by studying the structure of the collagen in your lectures, that the cross links between the collagen molecules are made by the covalent bonds. I hope that this MCQ is clear to all of you. Let's now move on to question number 7 over here. Question number 7 is a bit lengthy question and also a very tricky question. So let's now start with question number 7. Question number 7 over here says that the table shows some information about the polypeptides that make up the hemoglobin. So they are basically saying that the alpha globin chain and the beta globin chain. Alpha globin chain has 141 amino acids and the beta globin chain has 146 amino acid residues, right? Amino acid residues. Now they are saying that the position of the amino acid cysteine in the polypeptide chain. Now alpha globin only has one amino acid cysteine, right, which is on the position 107, and the beta globin chain has two cysteine amino acid which is on position 93 and 112. right? So now let's see that what they are asking us further in the question. They are saying that scientists studied the region of the beta globin polypeptide chain containing the amino acid cysteine at position 93. They found that this region faces outwards when no oxygen is attached to the heme group. This region faces inwards when oxygen is attached to the heme group. Replacing cysteine with a different amino acid residue reduces the Bohr shift. Bohr shift is in the oxygen dissociation curve. It is basically an S-shaped curve like this. इस तरह से कुछ like this, somewhat like this, इस तरह से curve बनता है for the Bohr shift, where basically the oxygen dissociation curve shift towards the right side. So let's see that what basically they are asking us in the question. They are basically saying that what can be concluded from the information about cysteine in the hemoglobin. Now they are saying that more than 1% of the amino acids in one hemoglobin protein are cysteine. तो अब हमें ये सबसे पहले we have to look at this that whether it is true or not. So in order to identify the percentage of the protein cysteine, सबसे पहले first of all we have to first look at that how many total amino acids are there in the hemoglobin molecule. There are 2 alpha chain and 2 beta chain. So we will multiply these values with 2. Right, so the answer we are going to get is 574 amino acids. Total we are going to get the 574 amino acids. And then over there they are saying that one alpha globin chain is having one amino acid cysteine, right? And there are a total of 2 alpha globin chain in a hemoglobin molecule. So over here we can say that alpha globin chain has basically 2 cysteine molecules, 2 cysteine amino acids, and the beta globin chain in total has 4. In total has 4 amino acid cysteine, right? Because there are 2 beta globin chain over in the hemoglobin molecule, and there are 2 alpha globin chain in the hemoglobin molecule. Alright, so the total cysteine molecules, the total cysteine amino acids basically in the hemoglobin molecule are 6, right? So now the how we can calculate the percentage of cysteine. Basically the percentage of the cysteine can be calculated by 6, the total number of the cysteine molecules divided by the total number of the amino acids in the hemoglobin molecule into 100 is equal to 1.05%, right? So the answer over here is option A. Yes, option A is our correct answer, that more than 1% of the amino acid in one hemoglobin protein are cysteine. We can clearly say that we can clearly see that it is more than 1% over here. Now we can also look at the other more relevant option, that in alpha globin chain there is a cysteine closer to the end of the polypeptide chain with an unreacted carboxyl group than in the beta globin chain. Now basically let's see that whether cysteine molecule is closer to the end of the polypeptide chain, or the cysteine of the beta globin chain is closer to the end of the polypeptide chain. We have a total of 141 amino acids in the alpha globin chain, right? And they are saying that the cysteine is found in the 104th position. So if we subtract 104 from 141, so basically our answer would be I guess it would be 37 residues if I am not wrong. Yes, 37 residues. So basically to reach the end it has the 37 residues of amino acids. And if we talk about the beta globin chain, so beta globin chain has 146 amino acids in total, right? And the closest amino acid cysteine to the closest amino acid cysteine to the end of the polypeptide chain is on the position 112. So if we subtract 112 from 146, we are basically left with 34 residues of amino acids to reach the end position, right? So basically in the alpha globin chain there are 37 residues left to reach the end position for the cysteine, and for the beta globin chain there are 34 residues of amino acid which are left to reach the end position. So basically over here they have given us the vice versa situation. So in the option for alpha globin it should be beta globin, and in place of the beta globin it should be alpha globin. Then this answer would have been considered to be right. I hope that this MCQ is clear to all of you. It was a little lengthy MCQ and also a very much tricky MCQ as well. I hope that this MCQ makes sense and it is clear to all of you. Alright, so now let's look at question number 8. Question number 8 over here says that which feature of the glycogen distinguishes it from the starch. Alright, so basically starch is made up of the amylose and amylopectin. And amylopectin is a branch structure, right? Amylopectin is a branch structure just like glycogen is a branch structure. But the basic difference which is between the glycogen and the starch molecule is that the amylopectin basically is less branched and the glycogen is basically more branched. So hence the answer over here would be option A, that all glycogen molecules are highly branched. The glycogen molecules are more branched than the starch molecules. If we talk about the glycogen molecules, it would be having more branch, and if we talk about the amylopectin molecule in the starch, it would be basically having the less branches. And also let's talk about some more differences as well, which is between the glycogen and the starch. They are that the starch is basically more compact. Sorry, starch nahi, the glycogen, I'm sorry, the glycogen is basically more compact and more soluble in water. More compact and more soluble in water than the starch. Alright, so I hope that this MCQ is clear to all of you. Now let's move on to question number 9 over here. Very simple and a easy question. Question 9 over here says that the diagram shows a biological molecule. We can see the ester bonds over here. These are the ester bonds, and we know that which biological molecules contain the ester bonds. They are the lipids. Lipids contain the ester bonds. So now let's see that what they are asking. Which molecule would be produced if this biological molecule was hydrolyzed? Basically we know that over here it is shown a triglyceride, and triglyceride is basically made up of the one glycerol molecule plus 3 fatty acid molecules, right? 3 molecules of fatty acids and 1 molecule of glycerol. This over here is the glycerol. This over here is the molecule of glycerol, and this over here are the 3 fatty acid molecules which basically combine together to give the triglyceride. So basically on hydrolysis what we are going to get, we will get fatty acids and glycerol only. So hence the answer over here would be option C, fatty acids and glycerol only. So I hope that this MCQ is clear to all of you. Let's now move on to question number 10. Question number 10 over here is saying that a mixture of glucose and starch solutions was placed in a length of the dialysis whisking tubing. We know that what is a whisking tube. Basically it is somewhat like this. It is a tube which is basically made up of the partially permeable membrane. It is somewhat drawn like this. Over here is the distilled water basically. Yeah, so now let's read the question, that what they are asking us in the question. And the tubing sealed. The tubing was then placed in a boiling tube containing the distilled water. 2 samples were immediately removed from this water. Time 0 minutes, and tested with either iodine solution or the Benedict solution. This was repeated at 10 minutes interval for the 30 minutes. The iodine solution gave a orange brown color each time. The table shows the results of the Benedict test. We know that the test for the Benedict test is basically for the glucose, and the iodine test is basically for the starch molecules, right? For the presence of the starch, and basically an orange brown indication shows that there is no starch available, right? Now they are saying that what may be concluded from these results. So basically they have put a solution, they have put a mixture of glucose and the starch in the whisking tube, glucose and starch in the whisking tube, and over here was the distilled water, and they tested the distilled water for the presence of the glucose and for the presence of the starch, and these were the results which are shown over here in this table, right? So let's read the statements one by one, and let's see which of them may be correct. The pores of the whisking tube are too small for the starch molecules to pass through. Yes, this is correct because we know that starch is basically a large polysaccharide, right? The starch is basically a large polysaccharide, and the whisking tube is only permeable to the small molecules, right? So this answer is this statement is basically also correct. That's why we can see that there is no presence of the starch, right? There is no presence of the starch in the distilled water. Glucose diffused through the whisking tube down the diffusion gradient. Yes, this statement is correct. And we can also see that from the results that the concentration of the glucose is increasing. Basically, as we go from blue, green, yellow to red, it means that the concentration of the glucose in the solution is increasing. It means that the concentration of the glucose in the solution is increasing with the time, right? So that's why the color change is happening. Red indicates the very high concentration of the sugar, glucose, right? Water diffuses into the whisking tube. Alright, so this statement, the evidence for this statement is not given in the question anywhere, right? They have not discussed about the water being diffused in or out of the whisking tube or not. They have just given the reference to the glucose and starch. So, and we have been asked that what may be concluded from these results. So we cannot conclude this, that whether water diffuses into the whisking tube or out of the whisking tube. Alright, so the answer over here would be option B, 1 and 2 only. Alright, so now let's begin with question number 11. Question number 11 says that which molecules are globular protein. If we talk about the classification of the protein, there are the fibrous protein and then there are the globular proteins. In the fibrous protein, the polypeptide chain does not fold, and in the globular protein, the polypeptide chain folds. Right? Right, so now let's go ahead with the question. The question is saying about the globular proteins where the polypeptide chains are folding. So we know that amylase, which is a tertiary structure, amylase is an enzyme and enzymes are usually of the tertiary structure. So it is a globular protein. DNA polymerase is an enzyme. It is also a globular protein. Hemoglobin. Yes, hemoglobin is also a globular protein because it consists of the coiled alpha globin and the beta globin chains, right? It consists of 2 alpha globin chain and 2 beta globin chain. So the correct answer for this question 11 is option A, 1, 2 and 3. All are the globular proteins. I hope that this is clear. Now let's move on to question number 12 over here. Question number 12 over here says that the initial rate of a reaction catalyzed by the enzyme was measured at various substrate concentrations. Which graph shows the effect of a low concentration of non-competitive inhibitor on a reaction? So basically यहाँ पे in this question they are asking us about the graph of the non-competitive inhibition. Alright, so we know that in the non-competitive inhibition what happens is that the Vmax decreases, right? The Vmax decreases and the Km which is the Mitchell's-Menten constant, it stays unchanged या फिर it stays the same, right? This is what happens in the non-competitive inhibition. And if we talk about the competitive inhibition, so what happened in the competitive inhibition? In the competitive inhibition, the Km increases and the Vmax remains unchanged, right? So we can over here identify the graph, that this graph is of the competitive inhibition, where the Vmax, where the enzyme and the substrate या फिर where the rate of reaction is maximum, right? Where the Vmax is maximum with and without the inhibitor, like it is unchanged. Over here we can say that without the inhibitor also the Vmax is this much, and with the inhibitor also the Vmax is the same. So Vmax is unchanged. So this is the competitive inhibitor graph. And when the Vmax decreases, which is over here and over here, but this is not the graph of the Vmax. This is not the graph which we have studied in the lectures as well. So I don't know which sort of graph they are showing over here. The graphs are always like this, the graphs which we have studied in the enzyme chapter, right? And the Km over here in this graph, we can see that the Vmax is decreasing, right? So this the graph for the non-competitive inhibition is the graph which is in the option D. Hence the correct answer for this MCQ is option D. Number 13 over here says that gout is a type of the arthritis in which small uric acid crystals form inside and around the joints. It causes sudden attacks of severe pain and swelling. The diagram shows how uric acid is formed from hypoxanthine catalyzed by the enzyme xanthine oxidase, right? So now over here they have given us that how hypoxanthine is attaching to the xanthine oxidase, and then xanthine is being formed, and then that xanthine combines with the xanthine oxidase, which is the enzyme, and then the uric acid is formed as a product. Gout can be treated by using a drug called aeropurinol, which has a similar shape to hypoxanthine, right? So this is basically the allopurinol, and they are saying that this drug can be used to treat the gout arthritis, which is similar in shape to the allopurinol, is similar in shape to the hypoxanthine, right? So which sort of inhibition is coming to your mind while reading this statement, the similar in shape? In my mind, the competitive inhibition is coming, in which the inhibitor and the substrate are of the same shape, and what happens is that the inhibitor binds to the active side of the enzyme instead of the substrate. Let's now read the question. Let's now read the leftover question. What can be concluded from this information about how allopurinol prevents the formation of the uric acid? Now let's read the options one by one and try to get the right answer. Allopurinol, it binds to the active site of the xanthine oxidase instead of the hypoxanthine, resulting in the reduced production of the uric acid. Yes, this is the correct statement. We have got our correct answer in the first giving statement, that it will bind to the allopurinol will bind to the xanthine oxidase active site and will prevent the conversion of the hypoxanthine to xanthine and then xanthine to the uric acid, right? And it is an example of the competitive inhibition. I hope that this MCQ is clear to all of you. Now let's discuss question number 14 over here. Question number 14 over here says that phospholipids are formed in a similar way to triglycerides. A sample contains 6 phospholipid molecules. The molecular weight of phosphate-ine is 95. The molecular weight of each individual fatty acid, they are talking about one fatty acid, in this sample is found to be 282 grams. Alright, the molecular weight of glycerol is 92. The molecular weight of water is 18. Alright, now they are saying that what is the molecular weight of the sample in grams per mole. So basically now let's start by discussing the structure of the phospholipid. In order to solve this question, you must know about the structure of the phospholipid, that phospholipid contains a hydrophilic head which has glycerol and a phosphate group in it, right? And the phospholipid molecule has the two fatty acid tails, and these fatty acid tails, when combined with the glycerol, it basically loses two water molecules, right? In the process of the esterification, we know that also in the formation of the triglycerides as well, triglycerides as well, when there is one glycerol and three fatty acids as well. So when they combine together, so what happens that whenever the OH group combines with the COOH group, so a water molecule is lost, right? So each bond of the fatty acid with the glycerol is losing two water molecules. Alright, so now let's sum up all of these components, the glycerol, the phosphate and the fatty acids, right? So now let's add it. I'm adding it over here, 92, which is of the glycerol. We will add it with the two fatty acids. If the one fatty acid is of 282, so if we multiply it by 2, because there are two fatty acid chains in one phospholipid molecule, so we will get a number of 564, right? We will get a number of 564, and then we will add one phosphate group which is equal to 95. So the total answer which we are going to get is 751 grams per mole, right? This is the total answer before the without the water loss. Basically we have calculated. Now we know that when the fatty acid molecule will combine with the glycerol, there will be a loss of one water molecule as well, right? So what will happen is that we are going to subtract uh, two water molecules, which is 18 plus 18 is equal to 36 from the 751. So the total water molecule, there are two water molecules that are being lost, which is equal to 36 grams per mole. So we are going to subtract 36 from 751. So the answer over here would be 715, right? This is the answer for the one phospholipid molecule, right? This is the answer for the one phospholipid molecule. And what will be the answer for the six phospholipid molecules? So for the six phospholipid molecules, what we are going to do is that we are going to multiply 715 by 6, and what the answer we are going to get is 4290 grams per mole, and the closest answer to 420 grams per mole is option B, which is 4182. So hence the answer over here would be option B, which is 4182. I hope that this MCQ is clear to all of you. That was all about the solution of question number 14. Now let's move on to question number 15 over here. Question number 15 over here says that which statement about phospholipids in cell surface membranes are correct? Statement 1 over here says that fatty acid tails allow most ions to pass through the membrane. We know that there is the molecule of the phospholipid is hydrophilic from the head and the hydrophobic from the tails. So basically the fatty acid tails are hydrophobic in nature. They are hydrophobic in nature, and hydrophobic means that they are the water fearing, right? Let me correct the spelling of the hydrophobic. The fatty acid tails are hydrophobic in nature, right? So they are water fearing, and also that they are non-polar, right? Hydrophobic can also be said non-polars, and we know that the ions are polar. So the fatty acid tail does not allow The fatty acid tails, in short, does not allow the ions to pass through. So this statement is also incorrect. That the fatty acids allow most ions to pass through. Fatty acid does not allow. Alright. Now let's look at the statement 2 over here. Statement 2 over here says that the hydrophobic tail point inwards facing each other. Yes, we know that the cell membrane is basically made up of the phospholipid bilayer, right? Bilayer means that they are made up of basically two phospholipid layers, right? One on the top and the other on the bottom. So these two tails are basically facing each other. So this statement 2 is correct. All polar heads face the cytoplasm. No, this is not correct. Not all the polar heads face the cytoplasm. If we look at this, so the phospholipid which has a layer towards the cytoplasm, its head is facing towards the cytoplasm. The other head is basically facing towards the extracellular environment, right? The environment outside the cell. So one phospholipid head is basically facing at the extracellular environment and one phospholipid head is facing towards the cytoplasm, right? Because the cell surface membrane is made up of bilayer of the phospholipids. So statement 3 is incorrect. Now let's move on to the statement 4 and let's see that what they are saying in statement 4. It says that the phospholipids helps with the flexibility of the membrane. Yes, this statement is absolutely correct, that this is the function of the phospholipids, that it helps with the flexibility of the membrane. Hence the answer to this question is option D, 2 and 4 only. Let's now have a look at question number 16. Question number 16 over here says that in an experiment, piece of onion epidermis, which is a plant cell, are put into 3 different concentrations of sucrose solutions P, Q and R. The pieces of onion are left for an hour and then examined using the low power of the light microscope. Each diagram shows one cell from the epidermis that was placed in each of the sucrose concentration. What explains the appearance of the cell in solution Q? Alright, let's interpret the question first. So I believe that over here the cell is swelled up. So this may be the condition where the cell may be in the turgid position, turgid state, where the water has gone into the cell, where the cell has gained the water. Right over here, because we can see that the cell membrane has basically detached from the cell wall. Cell membrane has basically detached from the cell wall. So this condition would be called plasmolysed cell. This is the plasmolysed cell, and this cell is basically undergoing plasmolysis, right, where the cell membrane has been detached from the cell wall, and this is due to the water coming out of the cell. Alright, this is because of the water coming out of the cell, and this is, I think, the state of the equilibrium, the Q1. The Q is the state of the equilibrium because it is not showing any abnormal state. Now let's look at the options and try to find out the correct answer. The concentration of the solution Q is equal to the concentration of the solutes in the cell sap. Basically, they are not talking about the concentration of the solution. They are saying that the concentration of the solution Q and the concentration of the solute. So it makes no sense. The cytoplasm has the same concentration of sucrose as the solution Q. So basically they are over here saying that the cytoplasm has the same concentration of the sucrose as the solution Q. This is also not very much evident with the given information, so we cannot conclude this statement as well. The water potential of the cytoplasm is equal to the water potential of the vacuole. No, we cannot conclude this as well from the given statements. The water potential of the cell sap is equal to the water potential of the solution Q. Yes, this is our correct answer. that the water potential of the cell sap is equal to the water potential of the solution Q. Why? Because we can over here see that there is no change in the shape of the cell and it is looking as a normal plant cell which is in the equilibrium state, which means that there is no gain or loss of the water from here. So hence the answer to question number 16 is option D. Now let's move on with question number 17. Question number 17 over here is saying that the electron micrograph shows the rod shaped bacteria. The actual length of the bacterium is 2 micrometer. Over here they are showing the actual length of the bacterium, which is the 2 micrometers. In the micrograph they are showing it, and the diameter is 0.5 micrometers. Over here this is the diameter and it is 0.5 micrometers. Assume that the bacterium is a cylinder shape. Basically they are saying that assume that the bacterium is cylinder shape, means that we have to consider this shape of the bacteria as a cylinder, right? What is the surface area to volume ratio of the bacterium? So as in the question they have asked us to consider the shape of the bacteria in their cylinder form. So what will happen? So we will have to find the surface area to volume ratio with respect to the cylinder, right? So over here we have the formula for the surface area of the cylinder, and what is that formula for the surface area of the cylinder? It is that area is equal to 2 pi r square plus 2 pi r h. Now we know that the r radius is not given to us in the question, so how we are going to calculate the r? Basically we know that the diameter over here is 0.5. So to calculate the radius, what we are going to do is that we are going to divide the diameter by 2, right? So we are going to divide the diameter by 2, which is 0.5 divided by 2, and the answer which we are going to get is 0.25, right? So the radius will come to be 0.25, right? The radius is 0.25. We will put the values. We will put pi as it is in the calculator, and the height would be the height would be 2 micrometers, right? And then the answer that will come will be 3.54, right? 3.54 micrometer cube. And then to calculate the volume of the cylinder, we are going to use the formula of pi r square h, which is the formula for the volume of the cylinder, right? So we will also put the values over here and we will get our answer as 0.39. So to calculate the surface area and the volume ratio, we will add the values of the surface area, which is 3.54, divided by the value of the volume, which is 0.39, and then we are going to divide it and get our answer as 9 divided by 1, which will be a 9:1. So hence the answer over here, or the answer we are going to get, is the 9:1. So hence the correct answer for question number 17 would be option D, 9:1. I hope that this MCQ is clear to all of you. Alright, so now let's move on with question number 18. Question number 18 over here says that chickens have 78 chromosomes in the nucleus of a body cell. There is just a simple body cell which are having 78 chromosomes in them. How many DNA molecules are there in a chicken body cell at the start of the prophase of the mitosis? Alright, so before the prophase begins, there is also the S phase where the DNA replication basically takes place. And because of the DNA replication in the S phase at the start of the prophase, the content of the chromosome has basically been doubled. Alright, so basically how many chromosomes we will be having is 78 into 2, which will be 156. So hence the answer to question number 18 will be option D, which is 156 chromosomes at the start of the prophase of the mitosis. Alright, so now let's move on with question number 19 over here. Question number 19 says that the photomicrograph shows the cell undergoing the mitosis. Mitosis may we have how many steps? We have the 4 steps in the mitosis, which is the prophase, we have the metaphase, we have the anaphase, and we have the telophase, right? These are the four stages of the mitosis. They are over here saying that which statement describes what will happen next in cell X. Alright, so basically over here I believe that this is basically where the chromatin threads are there and they are being condensed. So I believe this is a prophase stage of the cell, and next will come the metaphase. I believe that this is a prophase. Let's because this may not be the anaphase, this may not be the telophase, and this cannot even be the metaphase, right? Because in the metaphase the chromosomes are aligned at the center of the cell, right? So this cannot be the metaphase as well, and we cannot also see the spindle fibers, so this cannot be the anaphase as well, where they are being pulled apart. This cannot be telophase as well, right? So what let's look at the options one by one, and then let's see that what can be the correct answer, that what will happen next in cell X, right? So if we say that the spindle fibers break down and the cell prepares for the cytokinesis, no, this may not be, because this may not be the correct option. Why? Because first of all the chromatin threads will condense to form the chromosomes, and then they will align at the equator and then separate. So option D is incorrect. Let's now look at option C. It says that sister chromatids move towards the opposite poles pulled by the spindle fibers. No, first of all they will align at the center as in the metaphase, right? And then in the anaphase they will be pulled by the spindle. Chromatin coils up tightly and the nuclear envelope breaks down. We cannot see a nuclear envelope over here, which means that the nuclear envelope has already broken down. So this cannot be the correct answer. So hence the correct answer over here is option B, which is chromosomes line up along the equator of the cell and attach to the spindle. Right, I told you this is a prophase, which is the closest resemblance to the prophase according to the options which are given over here. So after the prophase, what will happen, that the metaphase will come, where the chromosomes will line up along the equator of the cell and they will be attached at the attached to the spindle, right? So that was all about question number 19. Now let's move on to question number 20, which is basically the last question of the part 1 of this video, and then we will be covering question 21 to question number 40 in the part 2 of the YouTube video, which I will be adding the link in the description, right? You can find the link in the description below. Now let's continue with question number 20 over here. Question number 20 basically over here says that which processes occur in the bone marrow cells that are in a mitotic cell cycle. Alright, so over here they are saying that phosphate groups bind to the ADP molecule to form the ATP. Yes, because mitotic cell cycle is an energy consuming process and it requires the energy from the primary energy source, which is the ATP, right? ATP is the primary energy currency of the cell, या फिर primary energy source we can say, right? So the statement 1 is correct. Now let's move on to statement 2. It says that bonds form between the nucleotides in a DNA strand. Yes, this is also correct. The bonds form between the adenine and thymine and guanine and cytosine. Right, between adenine and thymine 2 bonds are formed, and between the guanine and cytosine 3 bonds are formed. So this statement is absolutely correct. And then the next statement says that hydrogen bonds form between the tRNA anticodons and mRNA codons. Yes, this is also the correct statement. We know that when the tRNA molecule comes and binds with the mRNA codon, then only the amino acid is put into the polypeptide chain, right? So statement 3 is also correct. Hence the answer to question 20 is option A. All these statements, option 1, statement 1, 2 and 3 all are correct. So hence the answer to question 20 is option A. Alright, so that was all about this video. I hope that you guys enjoyed solving these past papers with me, and if you guys did, then do remember me in your prayers and send a lot of good prayers on my way. And do consider subscribing this channel as well. Your support and your prayers means a lot to me. Thank you so much, everyone. Have a nice day. Take care. Allah Hafiz.